Hi guys. Right, so in this video I'm going to talk about protein structure. It's going to be very quick, hopefully. I'm going to try and go through it quite quickly. We're going to go from amino acids all the way to quaternary structure. Okay, so let's begin. Right, so let's begin with the amino acid. Okay, so here we go. We're going to start off with the amino acid. Very basic. Central carbon, carboxyl group, hydrogen, and amino group. Okay? That R is just for display purposes. There are 20 to 22, depending on which book you read, different versions of the R group. So for the sake of simplicity, we're just going to call it R1. Different R groups can be indicated with different numbers, but remember, they do take 20 to 22 very specific different groups, okay? Otherwise called side chains. Okay, and they give each amino acid its kind of unique properties, but each amino acid also has these three in common, i.e. they're identical. Right, so this is the amino group. That's the carboxyl group. Okay, and that's our amino acid. Now, proteins fall into the group of molecules known as polymers. It takes many monomers joined up together to make a long chain, and that long chain is what we call the polymer, or in this case the polypeptide. The amino acid here is what we call the monomer. Okay, so it's the single unit that's joined many times to make the larger molecule. Okay, so in this case the amino acid is the monomer. Okay, so the monomer, then, how do they get joined together? So what we would have then is another amino acid adjacent to our first one. This might have a different R group, or it might have the same one, with carboxyl group and H there. Okay? So what's going to happen now is what we call the condensation reaction. You'll be familiar with that. Right? So let's just remove the details that we don't need anymore. Now when you're describing this, you know, I think it's pretty easy to kind of draw the diagrams and indicate it visually, but when you're describing it, there's a few things that you have to say. You have to say that the condensation reaction occurs between the carboxyl group of one amino acid and the amino group of another amino acid. When this happens, you get water formed, and that water is formed from those atoms right there. So this will form water. Actually, that's not how I have to draw it. the way I like to draw it is this. So the water comes off right there, H2O. And what you're going to then have is the carbon with one less bond because it's lost the bond to the oxygen. You're going to have nitrogen with one less bond because it's lost the bond to that hydrogen. And so, to satisfy the bonding requirements, carbon will form a covalent bond with nitrogen. Okay? And as you will see, what's produced is the peptide bond. Okay, so this is the peptide bond between the carbon and the nitrogen. And everything else, we just have to draw. Remember, we've lost that OH there, and we've lost that hydrogen from the amino group there. Okay, so I'll just draw this out. That's the carbon with the first R group or the side chain. That's my amino group that's not changed because it hasn't taken part in the reaction. Peptide bond, NH, carbon, and everything else is the same. Okay. Okay, and there we have our dipeptide. That's the peptide bond. I'll label these things. So that is your peptide bond. Okay. You might also see reference to something called a peptide group, and that is these atoms right here. Let's label that peptide group. Okay, very nice. So here we have the product of the condensation reaction. Remember, we produce the water as well. Cannot forget that, all right? Because atoms don't vanish into thin air. So if we've lost the OH group and the H, they've gone somewhere, and that somewhere is that water molecule over there. Right. What you have to then imagine is that this can happen many times. So there will be another amino acid next to this one. Again, the NH group will be there. Maybe this will be another R group, different one, hydrogen, carboxyl group. This will be lost. It's gone to form another water molecule, and there'll be a bond there, and it can carry on. Do you get the idea? Right? So we will end up with a long chain of amino acids joined by peptide bonds. Okay? Now what we're going to do is we're going to move on to the other stages of other levels of structure of the protein. So we're going to have to get rid of this stuff. Let's just, one thing that you need to know is when your amino acids are joined together in this way, what you have is, firstly, it's a polypeptide. So say if we had 4, 5, 6, 7, 8, 9, 10 amino acids joined in this way, what we would have is a molecule called a polypeptide. Yeah, that was a dipeptide. 3 would be tripeptide, 4 would be tetrapeptide, and so on, until when you got many, you call that a polypeptide. And most proteins will have hundreds, even thousands of amino acids joined together. Okay? So, right. That's the polypeptide, and that is different from which amino acids have been joined together. Okay? So any 100 amino acids that are joined together, that's a polypeptide. They're all polypeptides. Okay? But the primary structure is the specific sequence of which amino acids were joined together. So you must be clear about the difference between the two. Which amino acids are joined together, i.e., which R group was that? Which of the 21 or 22 or 20 was that? Which of the 20 was that? So which amino acids are joined together, that is called primary structure. And this is very important in the determination of the overall shape, but we'll come to that later. Okay? All we need to appreciate right now is that we join many amino acids together, it's called a polypeptide, but which amino acids we join together and the sequence of those amino acids, that's called primary structure. Okay? Right, we'll come back and discuss secondary structure. Right, so in this one, we're going to discuss secondary structure, and we're going to simplify that polypeptide structure a little bit, so that we can appreciate what's going on, you know, in the larger molecule. It's difficult to have all the details right there, and probably confusing as well. Right. So what we need to remember is that between is that you've got your amino acids, and I'll just draw them as like beads now on a string. Okay, you've got your amino acids, but in the middle of each one, what's joining them is, if we just do a quick review, if we zoom into that bond, remember it was the peptide bond between each amino acid, and remember the group was CONH. So between each of these amino acids, we've got this peptide group, and remember this peptide bond. Alright? And what we have to remember is that between each of these peptide groups occurs between every amino acid. Alright? And every amino acid's got an R group sticking out. Okay? Is that on the screen? Right. So every amino acid's got an R group sticking out, and between every amino acid there's this peptide group. Now the peptide group is identical, but the R groups are different. And depending on the primary structure, i.e. the sequence of amino acid, those R groups are going to be a different arrangement of the 20 amino acids, of the 20 R groups. Okay? Now some R groups can be charged, some R groups can be negative, some R groups can be positive. Some will be hydrophobic, some will be hydrophilic. Some will have a sulfur atom in them so that they can form disulfide bridges, disulfide bonds. So, you know, these R groups are variable, and because there's a different arrangement of those for different primary structures, it gives every single amino acid with a different primary structure different properties. But we'll come to that later. Anyway, so this is a peptide bond, a peptide group, and this oxygen has a slight partial negative charge, and this hydrogen has a partial positive charge. Okay? Now you know what's coming now because of that. Now, it's not possible for these bonds to kind of rotate themselves in such a way that this oxygen can be attracted to that hydrogen, even though they have opposite partial charges. Okay? You know, I don't know, it's like you not being able to lick your elbow, for example. Something like that, okay? However, if I asked you to lick your toe, that's something you might be able to do. You want to give it a try? So that's what we're saying. This oxygen, it can't hydrogen bond with, you know, that hydrogen, but if this chain was to loop around on itself, it could hydrogen bond with the hydrogen of a peptide group about 4 amino acids down the line. Okay, and that's exactly what happens. Okay, so what will happen is that this chain of amino acids will loop around like this, okay, and we'll draw this in the kind of background. Okay. We'll loop around. We can loop around like this, and what's happening here is that, actually let me draw it a bit differently. What would be happening here is that the peptide group here can hydrogen bond with the peptide group over there. Okay? And in the same way, this can happen every 4 amino acids. So maybe this one, yeah, the peptide group here can hydrogen bond with that peptide group there. Okay? And in this way, you will form a looped structure that is stabilized by hydrogen bonding between the peptide groups which seem to be aligning because of the looping. Okay? So that is the first level of structure in which there's some kind of folding happening. Okay? And this is one type of secondary structure called an alpha helix. Okay? Because of the direction in which it's turning, the alpha bit, and the helix because it's forming a helical shape. Alright, so the alpha helix is stabilized by hydrogen bonding, importantly, between the atoms of the peptide group. Okay? So there will be maybe an oxygen here, an oxygen which has got a slight negative charge, hydrogen bonding with the hydrogen of a peptide group 4 amino acids down the line. Okay, and this repeats itself. Okay, forming a helical structure. Okay, so the hydrogen bonds can be formed in that way, or excuse me, or what can happen is, instead of a helical shape, the protein, the polypeptide chain can fold back on itself like this, and this is called a beta sheet, beta pleated sheet structure. So what's happening here is that instead of the chain looping around and forming hydrogen bonds, it's kind of forming adjacent alignments of the polypeptide, and in the same way, because we've got oxygen here and possibly a hydrogen of that peptide bond, so we could have could draw the diagram out like this actually. We could have an oxygen sticking out of the peptide group over there. Could just draw it properly. Right, something like this, and over, down here we might have a peptide group such that the oxygen is there, and there could be a bond like that, and that could be happening here, that could be happening here, here, here. So again, another different type of secondary structure, but again, it's still the peptide group and the hydrogen bonding between the atoms of the peptide group that are responsible. But it's just a different arrangement of the polypeptide chain in order to make that happen. This is called a beta pleated sheet. Okay? Because if you looked at this side on, this would be coming out and up and down and up and down in 3 dimensions, and because it's repeated that way, if you looked at it side on, it would have a very kind of pleated structure. If I drew it in 3 dimensions, would look something like that. Okay? So that's why it's called a beta-pleated sheet. Now there's two versions of this. This is called an antiparallel because if we follow the direction of the polypeptide, first it's going that way, then it's coming back that way. So if you follow the arrow around, it would be that way. And then if we went again that way, it would be that way. Okay? So that way, back, and back again. So that's why it's called an anti-parallel beta-pleated sheet. It could be parallel, okay? So it's a little bit more kind of difficult to get your head around, but not impossible. So we could have a chain that goes like this, and then kind of is not involved in the sheet, but then it might be, and then it's kind of loose again, and then it becomes part of the sheet again, and so on. Okay. Now, if we follow the kind of path of amino acids, the directionality of the polypeptide chain, this time we've got that way, and then it went around, and then again that way, then it went back round again, and again aligned itself. Okay? So because we've got the part of the sheet now with chains going in the same direction, this time it would be called a parallel beta-pleated sheet. Okay? So that's the different kind of secondary structure, but remember, they are all stabilized always by hydrogen bonds between the atoms of the peptide group. Okay? R groups not involved yet. Okay? In this case, for example, in this sheet, the R groups are sticking out. There. There. These guys might be going down, down, and these guys will be sticking up, out, out. Okay, they're not involved. And similarly in the helix, right, peptide groups are holding that helix together, and the R groups are sticking out like this. Okay, they are not involved in the helix or the beta-pleated sheet. Okay, now let's move on to tertiary structure. So if we have a look at our polypeptide chain, so if we have a look at our polypeptide chain, this is what it started off like, okay? So it was just a chain of amino acids, but because of the secondary structure, we now have bits of it that are helix. Some of it might be anti-parallel sheet, some of it might be parallel sheet, other bits might be helix, and so on. Okay, so that's kind of how you might want to think of secondary structure. We haven't got a kind of fully folded protein here, but we've got bits of folding caused by the secondary structure, but it hasn't folded up in 3 dimensions. It's still more or less in a linear kind of arrangement. Okay? So, tertiary structure then. So remember when we said that from the helix and also in the sheet, the peptide groups and the hydrogen bonding is holding that part of the structure together, and the R groups are sticking out. And remember we said that there are 20 different types of R group. Different types of R group, and they all have, you know, different properties. So this could be R group 1, for example, this might be 2, this might be 4, this might be 4 again, this might be 4 again, and this might be R group number 20. Okay? 20 different varieties. And many amino acids along the whole chain. And it's this sequence of amino acids that gives its unique properties. And in the tertiary structure is where we see how. Okay? So let's just do a quick review of the properties that these R groups can take. Some of them will be ionic, okay, which means they will have a positive or a negative charge. All right. Some of them will be polar, which means they've got partial positive or negative charge. Remember, we always denote that like this: delta positive or delta negative. Okay, it's not as strong as a full positive or negative charge. They might be non-polar. Oh, sorry. With polar, remember, they always like to interact with water, so I'll denote that by this sign, hydrophilic. Remember, not like, I'm not going to say that. Interact with water, okay? Polar and ionic, they interact with water. I'll call that hydrophilic as well. Non-polar is different. Non-polar R groups are largely made up of carbon and hydrogen only. They don't have any polarity, they don't have any charge, and so they don't interact with water. So I'll give them this sign, hydrophobic. Actually, I could summarize it that these are hydrophilic. Not sure if I spelled that right. Right. So these are hydrophobic, non-polar. Okay. Largely made up of carbon and hydrogen. No charge, no partial charge either. And these are hydrophobic. Okay. And then we have some R groups that have sulfur in them, and they can form covalent disulfide bonds. Okay, so remember, you've got 20 different R groups, and the R group might be ionic, the R group might be polar, the R group might be hydrophobic, and it might contain sulfur. Depending on that, it can take part in different interactions. Okay? Now let's see what those are. Right, let's just take this off. Now, if you've got ionic R groups, an R group might be sticking out from somewhere, and it's positive, and if it comes into contact with an R group that is negative, they will form an ionic bond. Okay? You might have polar R groups, in which case it might be something, for example, like an OH group with an oxygen that is partially negative, and it might come into contact with another polar group. Maybe it's an amino group which contains a hydrogen like this, and the hydrogen obviously is partially positive if it's attached to a nitrogen, not always. And in this case, they might form a hydrogen bond between them. Okay? And the other important point is that if they're ionic or polar, they will interact with water. Which means that they're more likely to be found on the surface of the protein interacting with the cytoplasm. Okay? Interact with water. If it's non-polar, they form something that's, quite conveniently called non-polar interactions. Okay? Think of science teachers, guys. They don't really like to mix with other people, but they do mix with themselves. Okay? So, non-polar interactions. Don't interact with polar groups, don't interact with water. They like to mingle with each other. Okay, that's how they are stable. Okay, so I'll just make that point here. They don't interact with water. And therefore, you'll usually find them away from the surface of the protein at the core, away from water or the cytoplasm or whatever that watery environment happens to be. Okay, so just remember that, away from water. Okay, and obviously the sulfur containing groups may come in association with each other so that a disulfide bridge can be formed. Now this is a covalent bond, the strongest of them all. Okay, so I should have done this in order of strength, but this is the strongest. Yeah, so let's just call that number 1. Ionic is the second strongest. Hydrogen bond is the third strongest, and the non-polar interactions is the fourth strongest, i.e. the weakest. Okay, now when you're talking about maybe the effects of temperature and things like that, it is most likely these hydrogen bonds and non-polar interactions that are the first to be broken because of kinetic energy, because they are the weakest. Okay, covalent bonds, ionic bonds won't be broken in the case of increased kinetic energy, but ionic bonds may be disrupted by pH. Okay? Right, so these are the interactions. Now let's look at what kind of effect they would have on our protein chain that's still in its secondary structure. Okay, but bearing in mind that depending on which R groups are sticking out in which places, these different types of interactions could be formed. So, if I have my polypeptide chain, I've got a helix, and then I've got a bit of a sheet right here. I might have another sheet bit. I might have another helical bit. Okay? And remember that all along our polypeptide chain, we've got R groups sticking out. Okay? Some of them are polar, some of them are non-polar, some of them charged, some of them not charged, some of them contain sulfur. Okay, now depending on where they are, they can form different interactions. So say if we had a negative here and a positive here, right? What you might get is that, you know, because the chain is still kind of reasonably flexible, we might get random movement of the chain such that this positive might come close to that negative. If that happens, they're going to form an ionic bond with each other, and in that case, whatever movement was required to bring those two together, the chain is not going to unfold back because that bond is now stable. Okay? So that interaction between these two, you know, it might have come about just by chance, but once that they're close together, you can't, you know, unfold it. That part of the structure is now there. It's formed, it's folded, and it can't be unfolded. Okay? We might have, for example, maybe some hydrophobic, a lot of hydrophobic R groups right here. Okay? And we might have a lot of hydrophobic R groups over here. Okay? So, as part of, you know, maybe this part of the chain is a bit flexible, and they might just randomly come close to each other, but because they're stable in that interaction, remember, non-polar hydrophobic interacts with other hydrophobic. Because that interaction is stable, this part of the folding, the chain, is not going to unfold because that interaction is now formed. So this part of the chain might come around and interact with this part of the chain. Okay? We might have, for example, an SH group here and an SH group there, by chance. And maybe they will form a disulfide bridge, but in doing so, we're kind of forcing these two parts of the protein to come together. Okay? So can you see now, because of our 4 types of interaction, our polypeptide chain is going to be forced into folding back in on itself. So let's just see how that might look. So we have our helix, right? And because that positive is interacting with that negative, the sheet is kind of folded back up like that, right? And because that part of the sheet was interacting with that part of the sheet because forced by this disulfide bond, that sheet is now on top like this. And because that hydrophobic region was interacting with that helix is now right up against it like that. And suddenly, we have a three-dimensionally folded globular protein, okay? 3D shape, otherwise known as tertiary structure, due to interactions between the R groups, R group interactions, yeah? 4 types of interactions, and this shape is dependent, therefore, on the sequence of amino acids. All the way back, I'm going to remind you what we talked about earlier. This shape is now dependent on which amino acids we have in which order. If we didn't have this amino acid right here, with this positive group, with this positively charged group right here, we wouldn't have been able to form that interaction. Okay, so our protein, our polypeptide chain would have folded differently. If we had 2 negatives, if this was a negative, negative and a negative, that interaction would not have happened. If there wasn't an SH group here, and say if it was something else, like a methyl group, that disulfide bridge wouldn't have formed, and therefore wouldn't have caused the folding of that part of the 2 polypeptide chains in that way. Okay? And if these amino acids were different, it might not have been hydrophobic, it might have been hydrophilic, and in which case they wouldn't have folded in that way. Okay? So what gives the protein its final shape is the interactions between the R groups. And what makes sure that those R groups are in the right place is having the amino acids joined up together in the right order in the first place. Okay? And what makes sure that those amino acids are joined together in the right order, that's a different story. Remember, it's ultimately down to the sequence of codons in the mRNA, but what gave rise to that, the daddy of them all, the genetic sequence, the sequence of nitrogenous bases in the DNA, in the gene itself that was copied to make the mRNA, right? So, one final thing, sequence of amino acids comes from DNA, mRNA, join them up in the right order, then you've got secondary structure. Because you've got the R groups in the right place, they can form the different R group interactions to further fold the chain into a 3-dimensional shape. But the story doesn't end there, because say if you need more than one polypeptide chain, that's called quaternary structure. So we might have something else right here, and maybe another polypeptide chain that's got its own secondary structure, that's got its own tertiary structure. But maybe it takes these 3 to come together in order for this thing to have a function. That's the key point. Shape is the same as saying, or very important to, function. If you change the shape because you've got a mutation and you've got the wrong amino acid in the chain, you're going to change the shape, and therefore you might change the function. Either the function might be something new, or more likely it's not going to work properly. Okay? Or if you're designing changes, if you want your protein to be stronger, more resistant to temperature change, you might say, I'm going to stick a few more amino acids with sulfur in it. Maybe I can increase the chance of disulfide bonds forming, and therefore, even at high temperatures, my protein might not denature. Okay? So, shape is important for function. Function might depend on having more than one polypeptide chain, and in that case, that is what's known as quaternary structure. Okay? So, these three subunits together is called quaternary structure. The folding of this subunit individually, the folding of the polypeptide chain, that is called tertiary structure. Okay guys? So that, I think, wraps that up. What I'll leave you with is, I will leave you with that. You join up your amino acids, primary structure. That structure starts to fold into a helix or sheet, secondary structure. When your R groups start to interact with each other and form a three-dimensionally folded polypeptide chain, that's tertiary structure. And if you need more than one polypeptide chain to come together to form a functioning protein, that's quaternary structure. All right, guys? Good luck.